Hello, welcome to another screencast. This one is on carbohydrates, and we're going to be looking here at the characteristics of carbohydrates in general, being able to be comfortable with the terms monosaccharide, disaccharide, polysaccharide, what they mean, and then looking at the importance of carbohydrates, who cares about them. So first of all, we can just drop this down right here. The importance of carbohydrates is that they are used for energy storage molecules, but they also play roles in structural roles in cells, cell membranes, and will also play very limited roles in communication between cells in terms of glycoproteins on cell surfaces. So their big role, energy storage, some structural molecules, as well as then communication, but to a much lower extent than the first two right here. So on to carbohydrates. Carbohydrates are composed of sugar monomers that are called monosaccharides, and they are carbohydrates, the carbo part here for carbon, and then hydrate, water, sort of H2O here. So if we add water to this carbon, we get carbohydrates, and all carbohydrates have carbon, hydrogen, and oxygen in this ratio, right? CH2O. So one carbon to two hydrogens to one oxygen. There are different categories of these individual single molecules, the monomers. These monosaccharides, some of them are aldoses, some of them are ketoses. This is really not primary importance here, but we just want to point out that all of these things, glyceraldehyde, ribose, glucose, galactose, dihydroxyacetone, ribulose, fructose, with the exception of a couple, they all end in ose. So if you're at a cocktail party and you overhear people using words that end in ose, they're probably talking about carbohydrates. The most familiar ones to most of you are probably fructose, probably not ribulose. Ribose is probably familiar to most of you. Glucose is definitely familiar to most of you. And some of you, galactose might be similar to you or familiar to you. Now, regardless, if you go through and count up here, you're going to see that the ratio of carbons to hydrogens to oxygens is in this CH2O here. So the empirical formula for these carbohydrates is CH2O. Now, these monosaccharides, these monomers, the single unit, single molecule here. This is what they look like in a chain form right here. So the carbons are all in a linear chain. But most often you're going to see the most famous of these monosaccharides, glucose, in its ring form. So here's the ring form. I want to just point out to you that you can build this in class with the three-dimensional models, and it'll make a lot more sense to you if you come and build this thing. So this is, again, glucose is C6H12O6. Right here are the 6 carbons. Right? One is up off the ring here, and then one of the oxygens is in the ring. But the basic shape here is a hexagon. Right? Now the image down here is they shorthand this a little bit, and they remove the carbons, and it's assumed that each vertex there is a carbon here. Right? And this is a little bit wider and fatter here for a reason. It's supposed to show you that the plane of the ring is in a plane that's coming out at you. And again, building this in class with the 3D models is going to be really beneficial in order for you to see that. But you being able to recognize sort of hexagons as monosaccharides, glucose is an example of that, is going to be key here, because you're going to see a lot of hexagons. When you see hexagons in our textbook, they're probably representing glucose, a monosaccharide. Now, to form disaccharides, you are going to take 2 monosaccharides and condense them together. So this is going back a few screencasts where we talked about dehydration reactions, condensation reactions. Well, here we go again. We're going to take glucose and condense it to another glucose. So we're going to take the OH group here. We're going to take the hydrogen from this hydroxyl group here, bring them together to get a water molecule. We're going to break this bond, break this bond, and we're going to bring these electrons over here and make a bond right there. And so what you get is a glycosidic linkage, this 1,4 glycosidic linkage, because it's between carbon number 1 and carbon number 4. If you go back a slide, just real quick there to bring these numbers out, they number the carbons, they, biochem nerds, number the carbons in order to keep track of them. So this is always carbon number 1 next to this oxygen. This is carbon number 2, this is carbon number 3, carbon number 4, carbon number 5, and then carbon number 6 is up off the ring there. This actually will be important later on when we do DNA, so we're going to come back and see this. So it is worth your time to sort of take a note, make a note there that the carbons are numbered. So we get this 1,4 glycosidic linkage here and this condensation reaction to get this thing called maltose, which is the sugar in Whoppers, the candy that is handed out at Halloween that no one really eats, at least not in my house. Maltose here is a disaccharide, and it is made up of 2 individual glucose molecules. But maltose is not glucose. Maltose is maltose. Has different chemical reaction properties, different chemical properties than glucose alone. So if we come down here and look at a dehydration synthesis of sucrose, we're going to take a glucose and a fructose here. Now, glucose and fructose, they both have the formula C6H12O6, C6H12O6, but they are isomers of each other, isomers of one another. They have the same the same molecular formula, but different structural formulas. You notice that this is a hexagon and this is a pentagon, even though that there are still 6 carbons here, carbon 1, 2, 3, 4, 5, 6 off the ring there. So you've got these 2 carbon things that are off the ring in a fructose molecule. Fructose tastes sweeter on your tongue than glucose does because of the structure. Moving on, we're going to condense these things together right here and get another, to get this, get a 1,2 glycosidic linkage here, and make something called sucrose, which is, again, a disaccharide, and this is table sugar. So this is one molecule, sucrose is a molecule, maltose is a molecule, but these are disaccharide molecules compared with glucose and fructose, which are monosaccharide molecules. So the sucrose and the maltose are physically bigger, larger than the glucose molecule or the fructose molecule individually. Moving right along here, we can now make polysaccharides, and we just want to show you a couple of examples of polysaccharides here. Again, we're going to do this the same way that we made a disaccharide, by condensing monomers together. But there are a couple differences here. One, actually, let me come back to this slide. Sorry, these are a little out of order. Let's go up to this slide here. Polysaccharides in plants, right, are generally called starch. So these are plant polysaccharides. And there are 2 flavors of them, amylose and amylopectin. So amylose, again, here are the little hexagons, right? So hexagon, 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 polysaccharide, made of individual units, monosaccharides. The amylose is in a sort of a straight chain, and the amylopectin sort of branches there. So plants store any extra glucose they make as starch. So if you see, here's a chloroplast and the starch granules inside that chloroplast all summer long. When primary production is pretty high, they're making a lot of organics, and they don't use as many of those organics in respiration as they're making during the long days of summer. So they store them, right? They store any extra organics here as starch, or any carbohydrate organics as starch. In animals, if we consume lots of glucose, one thing that our cells will do is store it as a polysaccharide known as glycogen. So this is how animal cells store extra carbs, extra glucose that comes in. So glycogen is stored in the liver and is also stored in muscle cells. So similar but different. Now, what's the difference between starches and glycogen? Well, you see the glycogen much more highly branched. Right? We have branching connections here, right? Branching connections, right? Etc. So it's a little bit more like the amylopectin than it is like the amylose. Right? So polysaccharides there. We need to go back and just talk about a couple other polysaccharides in the plants, right? So animals, pretty straightforward, just like glycogen, but in the plants, another polysaccharide in plants is a structural polysaccharide called cellulose. So here's the cellulose molecule, and it's in the cell walls of plant cells. And these cellulose microfibrils in the plant cell wall, if you pull them out and zoom in on them like this, there are many, many chains of these polysaccharides right here. And so each individual monosaccharide, and then you get these cross links here, cross links between the individual between the individual polysaccharide chains. And so if you look here, cellulose is made up of beta glucose monomers. Right? Now, I apologize for this being out of order, but we need to just go back to a couple of slides here. So you have beta glucose and alpha glucose. And this is like second tier information, but it's important if you're trying to understand why starch is different than cellulose, and it has to do with the glucose that's there. So starch is all glucose, cellulose is all glucose, glycogen is all glucose. Just the difference in plants and animals, right, for the starch and the cellulose versus the glycogen. But in plants, again, we're looking here between the alpha glucose and the beta glucose. If you have beta glucoses, it all goes back to this carbon number 1 right here. This hydroxyl group is in an up position above the ring, and again, it'll be helpful if you build this in class if you really want to see this. So this is in the up position, but on carbon number 1 over here, the hydroxyl group is in a down position below the ring. So an alpha glucose is down, and a beta glucose, the hydroxyl group is up. And you're like, what is the big deal with this? The big deal is that beta glucose is when you string them together, right, you end up getting cellulose, right? But alpha glucose is when you string those together, you end up getting starch, either amylose or amylopectin. Okay, so the difference is right here. So kind of spelling, if you will. Spelling does matter. Where you put this hydroxyl group changes how this molecule is going to react, changes how this molecule is going to react to one another. So the final slide here is, again, just showing the difference between starch and cellulose. So starch is a 1,4 linkage between alpha glucose monomers. And what happens is you get all these carbon groups that are up above this ring oriented like that, whereas in a beta glucose, because of the way that it just bonds together, you get this alternating, right? And that alternating is what aids in making the cross links. Right? These cross links prevent dissolving, and also sort of prevent any sort of breakdown. We, as animals, right, humans, don't have the enzymes to break these 1,4 linkages of beta-glucose monomers. So when you eat a lot of plant material, you're not able to break down that cellulose. You're not able to digest it because you don't have the enzymes to do that. And so that cellulose just sort of passes through, and this is what is known as giving bulk, or is what is known as fiber, right, in your diet. This helps keep you regular, helps keep your intestinal tract moving and pushing stuff through there. So we do not digest cellulose. We do, however, have the enzymes to break the 1,4 linkages of the alpha glucose monomers here. So we do have the ability to break down and digest starch in our diet. Just some interesting facts. And that brings us to this summary slide here, which is helpful for you to kind of organize your thoughts again there and complete that. So questions, write them down, bring them to class. Hope that was a little helpful.